Please join me in welcoming Dr. Carolyn Parkinson. Welcome. Thanks for joining us tonight.
Hi there. I'm just getting my screen sharing set up. So thank you so much for inviting me to give this presentation, and I'm really glad that you guys could all join us tonight. The topic that I want to tell you about is twofold. First, what can we learn about the brain from studying our social networks? And second, what can we learn about our social networks, so the social connections that we have with other people and those that we might be likely to form in the future from studying our brains? So before we get started, I wanted to make sure that I was really clear about what I mean when I use the phrase social networks. And I think that this is a really important concept to clarify early on in the talk, because when a lot of people hear the phrase social networks, their minds automatically jump to only social networking websites or apps. But social networks don't just encompass things like online communities where you might follow people or become virtual friends with them. So things like Twitter, Facebook, MySpace some time ago now, and Instagram and things like these. Although researchers sometimes use data from websites and apps like these to conduct social networks research, generally this is not all that's meant when we talk about studying our social networks. Instead, we're usually talking about the kinds of social networks involving oftentimes face-to-face interactions that humans have been making for thousands and thousands of years. So these go back a long way. In fact, the kinds of social networks that humans build and inhabit, they don't just predate the internet, they're also thought to predate agriculture. So there's some really interesting work looking at social networks in hunter-gatherer societies like the Hasda pictured here that are thought to resemble in many ways the kinds of societies that pretty much all humans inhabited tens of thousands of years ago. And many characteristics of the social networks observed in these groups closely mirror the kinds of social networks that we create and inhabit and the kinds of societies that you and I likely live in. So I've been told a lot of the audience today is from the United States, so highly developed, industrialized societies. So human social networks as they are today don't just predate the internet, they're also thought to predate things like agriculture. And you can also study social networks in other species, like in dolphin populations, for example. So people have looked at what dolphins associate with one another and which ones don't associate with one another or avoid one another, and then studied things like how behaviors spread in dolphin networks. And so the research on the neuroscience of social networks that I'll be telling you about today is not about hunter-gatherer bands or about communities of dolphins, but the reason that I'm giving you these examples is just to make the point that social networks are not just bound to online apps or websites. So no matter what culture or location or era you're living in, you're going to have some set of family members, friends, coworkers, and neighbors who each have their own sets of family members, friends, coworkers, and neighbors. And as a result, we form social networks where we're connected to a number of people in a particular set of ways. And all our thoughts, all our feelings, and all our behaviors are embedded in our social networks. So hopefully that clarifies what I mean by social networks in the title of my talk. And then going back to this title, at first glance, this combination of topics, brains and social networks, might seem a little odd or incongruous to you. So you might be thinking, well, first, why on earth would we think that studying our social networks would help us understand anything at all about the human brain? Well, one reason is that we seem to spend a lot of time using our brains to think about other people and where they sit in our social networks. So one window into the kinds of things that people spend their time thinking about is the kinds of things that people spend their time speaking about. And by some estimates, about 2/3 of what we talk about revolves around other people and the social interactions and relationships between those people. So essentially, gossip, broadly defined. And so a lot of people use the term gossip in a derogatory light, but as humans, we seem to be quite inclined, for better or for worse, to keep track of who's a friend of whom, who's on the outs with whom, who's in a position of power, and the content of our conversation certainly reflects that. And you can also think of some of the very first conversations that you might have when meeting someone at a party or a wedding or something for the first time. So in a lot of cases, you might spend that first conversation with a new acquaintance trying to map out how they might be connected to you socially. So you might say, oh, how do you know the hosts? Or you're from Kitchener. Oh, my friend Amy grew up there. Do you know her? Or, oh, I have a friend that works at that company. Do you know Mike? And so on. And so we often spend our very first conversations with people trying to map out the social connections that tie us together. And if you're like most people, you might even get a feeling of real satisfaction if you succeed at doing this. So we seem really driven to do this. We like doing this. People even play games like the 6 degrees of separation game, or sometimes the 6 degrees Kevin Bacon game, to try and map out the social connections between people as a pastime because it's so satisfying. And in fact, just as an interesting side note related to this, the observation of this tendency to use our first conversations with people to map out how they might be indirectly connected to us through chains of social ties inspired some early research on social networks, which was done in part by the social psychologist Stanley Milgram, who you might know from his famous work on obedience. So he and his colleagues noted that people often use those first conversations to try to figure out how they're connected to each other, and then often end up marveling at how small the world seems to be. And he wanted to see how small the world really was, even when you can't successfully map out the social ties that indirectly connect you to somebody. And so he did some research trying to map out how many degrees of separation it takes to connect people within the United States. And that's where that phrase 6 degrees of separation comes from. And so those studies were really inspired by this observation that we often spend the start of our time with new people mapping out the connections between us. And so we spend a lot of time speaking about our social connections. It also turns out that even without our awareness, our brains also spend a lot of time helping us prepare for social interactions and generally processing social information. And so you can also look at what happens in our brains when we're not doing very much at all. So the regions that you see here are part of what's called the brain's default mode network. So on the lateral, or rather on the left side of the slide, is a lateral view of your brain. So if you were to look at someone from the side, and on the right is a picture of what it would look like if you took that same view, so looking at someone in profile, but examined a slice sort of going right down the middle between their eyes, in other words, a medial view. And the default mode network got its name because those areas are more engaged when people are resting. So when they take mental breaks between tasks. And it was later found that these very same brain areas are really reliably activated when people think about themselves and think about other people. So that raises the question, is this just a coincidence, or is this neural activity at rest, which happens in the very same areas that support processing information about other people? Is this playing some role in preparing us for social interactions? And some really fascinating work by my colleague, Matt Lieberman and Megan Meyer, has shown exactly that. So when people activate these regions more during a mental break, if you give them a task right after that break, they're better at it if it's a social task, like inferring what someone might be thinking or remembering things about other people. But this doesn't hold true for non-social tasks, like remembering things about physical locations in space, for example. So the overlap shown in these pictures doesn't seem to be a coincidence. The pattern of activity that our brains tend to have at rest actually seems to play a role in helping us to gear up to effectively understand and interact with other people. So we seem to spend a lot of time processing information about other people and the relationships between them. We also seem to be really good at this. And so one of the most challenging sets of tasks that we're faced with every day involves what I just described. So figuring out other people and the relationships between those people, the social lay of the land, so to speak. And this allows us to think and act appropriately in various social situations. And typically doing this doesn't make us feel a sense of much effort or exertion, even though when you step back and consider it, it's a hugely demanding and complex cognitive feat. So to illustrate this, I want you to consider what would have been an unremarkable everyday experience pre-COVID, and hopefully will be soon again. And that's just encountering someone, bumping into someone you might know on the street. So when you encounter someone out in the world, how you respond to this person, so how much attention you might pay to her, if you stop to say hello, the kinds of things you might say if you were to stop to say hello, even things like how much you might trust this person, these things all depend in part on aspects of her as an individual. So things that you'll pick up on and process or recollect about her appearance, her personality traits, your knowledge of her likes and dislikes, how you remember prior interactions with her. And then in addition, how you perceive and interact with her is also going to depend on your relationship with her. And so one aspect of this is how close you are to her. So whether she's a close friend, close acquaintance, or friendly stranger. So in addition to characteristics of people in isolation that can shape our behavior, there's also an understanding of how close this person is to you. So are they in your inner circle, further out? Maybe these people are acquaintances, maybe they're practically strangers. But it also gets more complicated than that, because it's not just that people are closer or farther to us in one dimension and that's it. We each have our own sets of social connections who in turn are connected to each other and to others. So we're embedded in this rich fabric of social ties. In other words, our social networks. And as you'll see throughout my talk, information about someone's position in our social networks seems to matter for our behavior. So some people may be better connected than others, for example, and now we might have to modulate our behavior accordingly, because now there might be more pronounced reputation costs for doing things like divulging sensitive information or wronging them or something like that. So as humans, we seem to be extremely adept at seemingly effortlessly keeping track of a whole host of information about other people, and then using that information to guide how we think and how we behave. And another reason that we might learn a thing or two about the brain by considering our social networks is that the evolution of the human brain may be closely tied to the social networks that we, and of course our brains, are embedded within. So some people, like the anthropologist Robin Dunbar, for example, have prominently argued that the cognitive demands of doing things like keeping track of all this information about other people and modulating our behavior accordingly might have been a driving force in human brain evolution. And this idea is known as the social brain hypothesis. And you can see that humans have unusually large brains for our body size by looking at this graph, for example, where brain weight is on the y-axis and body weight's on the x-axis. You can see the slope of this line summarizing the relationship between these variables in vertebrate animals is quite positive, which shouldn't be altogether surprising. Bigger bodies need bigger brains, even just to control them motorically. So you get a bigger brain with a bigger animal. The surprising thing is that humans, or man as we're labeled in this graph, we're pretty far away from this line and we're quite a bit above the line. And so our data point is way above this general trend line, meaning that the actual value of our average brain weight is greater than the predicted value based on our body weight, which would correspond to the where the y-axis value for our body weight would fall on this trend line. So you can see our dot is above the line. And other species above the line, so other species who have bigger brains than you'd expect based on their body size, include some of our primate relatives and other animals like dolphins, for example, that have quite complex social lives. And consistent with the idea that our outsized brains for our body size might have something to do with our big complex social worlds, if you look within primates, for example, you see that primate species with larger brains for their body size tend to live in bigger social groups. And the relationship is particularly strong if instead of looking at brain size, which is an extremely coarse measure, you look at a very slightly less coarse neural measure, and that's relative neocortical volume for one's brain size. And neocortex is just the most recently evolved component of the cerebral cortex that's responsible for a lot of higher order mental processes. And you can see that this positive relationship emerges in this graph, where average social group size is on the y-axis and relative neocortical volume is on the x-axis, consistent with the suggestion that much of what's distinctive about the human brain, its unusual size and the impressive cognitive abilities that it affords, might have evolved so that we can keep track of and navigate our large, complexly bonded social groups. So in order to do things like accrue advantage and manage reputations and seek and spread information effectively and avoid conflict, we need to manage and remember many different relationships between other people and shape our thoughts and behavior accordingly. And this is all really computationally demanding and requires a lot of brain power, to put it in really, really coarse terms. It's also worth noting that if you take relatively young kids, so kids about 2.5 years old before literacy and schooling typically kick in, and compare their cognitive abilities to those of our close primate relatives like chimpanzees and orangutans, we don't really stand out when it comes to non-social cognitive skills, things like spatial memory or quantity discrimination, skills that would be useful for things like finding food, for example. But where we really shine is our social cognitive abilities. So things like learning by watching others and communicating and paying attention to what other people seem to be thinking, like where they seem to be looking, for example. So understanding other people and the webs of social connections between other people is something that we seem to be really adept at. It's something that we seem to be concerned with nearly all the time. And it's something that seems to have been likely to have shaped the evolution of our brains. So I think that it's something that's really worth trying to understand, but we still know very, very little about how our brains do this. And this is in part because, with a few exceptions, attempts to study the social brain have historically involved people laying all by themselves in noisy tubes like this MRI scanner that you see pictured here, doing things like looking at pictures of strangers. So in a typical study, we would tend to show someone some generic stimulus set. Like, for example, if you were a participant in a study like this, we might compare how your brain responds to the stranger smiling versus the stranger scowling. So it's rare that we would take real world social relationships into account. And when we do, we tend to look at very crude things like compare how your brain responds to someone who you know. So if we knew each other, that could be me, versus someone who might not be personally familiar to you, but might be visually familiar to you. So someone famous like Justin Trudeau here. And these kinds of studies can tell us a lot, and they have told us a lot. So for example, seeing a personally familiar face will engage brain regions involved in interpreting social cues more than seeing a stranger would. But not a lot of research has really gone beyond these relatively coarse distinctions between friend versus stranger, for example. And if we constrain ourselves to only looking at these stripped down comparisons and paradigms, we risk missing out on much of the picture when we're trying to understand the neural underpinnings of social thought and behavior. So in general, the fields of individual psychology and neuroscience and of social network science have until recently really been siloed. And this has recently started to change. So there's been a real groundswell of people bridging these disciplines by using data from people's real world social networks to gain insight into the structure and functions of the human brain. So I'm going to start by telling you about some of that research and what it's taught us about the brain in the first part of my talk. And then in the second part of my talk today, I'm going to tell you about the other side of the coin. So how we can learn about our real world social networks, for example, predicting friendships between people by studying our brains. So to answer questions in this first category, what can social networks tell us about the brain? We've done a variety of studies where we've asked questions such as, do we have accurate knowledge of where other people sit in our social networks? And do our brains spontaneously activate that knowledge when we encounter those people to shape how we think and how we'll act? And there's reason to think that this might be the case. So as I alluded to at the start of my talk, other aspects of our knowledge about familiar people, like personality traits, for example, are thought to be spontaneously activated by the brain when you encounter someone. And the purpose of activating this knowledge is thought to be preparing us for adaptive and effective social interactions with them. So this allows you to sort of seemingly effortlessly shift gears depending on who you happen to find yourself around. And information about social ties, like whether someone's a friend or a friend of a friend, or characteristics of their position in your broader social network, like how well connected they are, these things also likely inform our behavior and our perceptions, as I mentioned earlier. But as I also mentioned earlier, while there's been a fair amount of research contrasting things like neural response to friends and strangers, with a few exceptions, there really isn't much known about if and how we encode and are affected by specific social relationship information about familiar others. And yet these are the people, so our friends, our coworkers, and so on, with whom most of our everyday interactions take place. And so to begin to get at this and work with Adam Kleinbaum and Talia Wheatley, we took all the first year graduate students in an academic program at a rural isolated university, so about 300 people who generally just eat and live with and study with and socialize with one another, so sort of a bounded community, and we reconstructed their social network. And we did this simply by asking them to answer this survey question about who they socialize with most often as part of a classroom activity that they didn't know would be related to the fMRI study they'd be recruited for a few weeks later. So the prompt read, consider the people with whom you'd like to spend your free time. Since you arrived at this institution, who are the classmates you've been with most often for informal social activities like going out to lunch, dinner, drinks, films, visiting one another's homes, and so on. So this is how we defined friendship for them, so it would be understood consistently for everybody in the study. So regular socializing partners. So students essentially just name their friends, and then using this information, we could reconstruct the social network of the cohort. So all the circles here are students, and all the lines are friendships between those people. And doing this allows us to map out the structure of these students' social worlds. And then we had some of these people, so the circles that you see colored in peach here, come in for an fMRI study, where we could look at what happened in their brains when they saw one another. And specifically, we want to see if their brains would encode their classmates' positions in their social networks when seeing one another. And the properties of people's positions in such a large network are pretty hard to visualize here, especially when showing all the students and all the relationships between them. Kind of just looks like a big ugly hairball or something. So I'm just going to quickly go over some of the key aspects of social network position that we're interested in using a simple toy network of 13 people where they're easier to see. So one important consideration is social distance from you. And this is really easy to get at. It's just like playing that 6 degrees of separation game. So for any participant, you can take Mary, for example. We can see who's 1 degree away from her, like Tim, who's 2 degrees of separation from her, like Luke. And we can play this game for everybody in the network and map out how far or close each person is from Mary. And you can imagine how this information might shape Mary's behavior. So the actions and thoughts and feelings of people closer to her could be more relevant to Mary and more relevant even than those of people who aren't directly connected to her or people in her immediate social circle. So Mary might be more likely to tune into those people. And we also can extract other important aspects of people's social network positions from the patterns of social ties in this network too. For example, how well connected someone is to well-connected others. And this might be something that's especially important for humans because for us, our power to influence other people and things like conflict resolution and resource allocation depends not just on who's strongest or who's most dominant, or even who's most prestigious or who knows the most. If somebody has a lot of people who support them, who in turn have a lot of people who support them, they gain a kind of social power or status. And so we defined status here in terms of how well connected someone is to well-connected others, or eigenvector centrality. And the people high in eigenvector centrality are big fish in big ponds socially, essentially. Like Bob here. So even though Bob and Tim both have 4 friends, Tim's status in this sense is lower because his friends aren't as well connected. And this is similar to how Google decides what pages to rank first when you do a web search. So a link coming from my web page to your web page isn't as valuable as a link from like the New York Times coming to your web page. And similarly, Bob's link to Leah is more valuable to a status than Tim's link to Amy. And a third important aspect of social network position that we can extract from the patterns of ties in this network is who connects people who wouldn't otherwise be connected in the network. Or in other words, who serves as a social bridge or occupies a minimally constrained network position. So you can think of these people as sort of social bridges, or sometimes they're called social brokers. So you could consider maybe someone like Tim here, who in high school might have been the kind of person who would be friends with people spanning different cliques. In contrast, someone like Lisa here only has friends who are already friends with one another. So compared to Lisa, Tim will tend to get exposed to more diverse information and ideas. He's also more likely to be something of a social chameleon who adjusts his behavior depending on his social context. So we can characterize everyone in terms of how constrained they are in the network. We all vary on how well connected and how constrained we are in our social networks. And we are, of course, closer to some people than we are to others. And together, these describe much of the structure of our social sphere. So we can take everybody in our network and characterize them in terms of these attributes. And this allows us to get a rich set of information about these people without having to ask them much beforehand, because we just ask them who they hang out with one by one, and without having to really clue them in on what the future study is going to be about. So as I said before, we took several people out of this network, the nodes that you see colored in peach here, and we had them come in for an fMRI study. And in this study, we wanted to see if our participants' brains would spontaneously encode knowledge of one another's positions in the social network when seeing them appear on the screen, which you could think of as analogous to how your brain might react when seeing their faces when encountering them on the street or at a party or in a classroom. So in an fMRI study, in case you're not familiar with how this would work, someone would lie in an MRI machine like the one you see here in the corner on the slide, and then we can show them different kinds of pictures or movies or sounds, or even have them do different puzzles. Here we showed them videos of various classmates, and we can see how the flow of oxygenated blood through the brain changes. And what you see here is an animated statistical map of changes in that signal over time, with hotter colors indicating more oxygenated blood flow. And throughout this talk for concision, I might say activity sometimes when talking about fMRI data. And what I'm really talking about when I say that in the context of fMRI is just changes in oxygenated blood flow. And if you heard about the results of fMRI studies in the news or something before, chances are they were based on pretty coarse analyses that historically have been the most common and that have focused on seeing what parts of your brain light up, so to speak, when doing different tasks or seeing various things. So in other words, how much a given brain region responds when doing a certain kind of task or when perceiving certain kinds of information. But over the past several years, scientists have started to leverage newer statistical approaches to take a more nuanced look at the distributed patterns of activity within brain areas, rather than just looking at how much they respond overall. So with these newer approaches, we're essentially extracting fine-grained distributed patterns of activity or neural signatures or response topographies, in other words, for seeing different things, and in this case, for seeing different people. So to hopefully clarify what I mean by this, imagine that this brain area that I've circled here in this cartoon depiction responds more to seeing these people who you know compared to some baseline, like for example, seeing the faces of strangers. And I'm going to use cartoon faces often on these slides to preserve our participants' anonymity, but imagine these are photos of real people who you know. So in addition to looking at how much this area responds overall when seeing these people, we can also take a closer look inside the brain region and look at the spatial patterns or topographies of activation depicted on the bottom row here. And we can ask, inside a brain region that might light up, so to speak, when seeing anybody who you know, is there information in these finer grained response patterns about not just the fact that you're seeing someone who you know, but also about their relationship to you and their relationships with one another and where they sit in your social networks? And so you could think of this approach as analogous to looking at something like different mountain ranges that have the same average height, but different profiles of peaks and valleys. So here we want to examine the patterns of activity evoked in different areas of the brain when seeing different people, and then compare those patterns to each other. So you can imagine that these circles with faces in them that you see here signify different neural activity patterns evoked in some brain region when seeing different people from your social network. And they're arranged such that faces that evoked more similar patterns are closer together in space. And in this example, I've also visualized how close these people are to you in social ties in terms of their color and how well connected they are, or how central they are, in other words, by their size. And so looking at things in this way, so comparing neural patterns evoked by different people to each other, this can help us to understand what role the brain region plays in processing information, or in other words, what distinctions it cares about, what rules govern, what it treats as the same, what it treats as different, and by how much. And you can think of this as sort of an information signature, so to speak, of that brain region. And in our study, we looked at what patterns of activity in your brain look like when seeing the faces of different people in your social network. And since we know about all the relationships between those people and can extract their social network position information that I told you about earlier for everybody that each participant saw in the fMRI study, we can test if and where the local information signature, so the way that a particular brain region divides people up or bins people when you see them, reflects distinctions based on how many degrees of separation they are from you in your social network, which would look something like what you see here, where the people who are close to you in social ties are all close together in space, and far away from people who are a few degrees away from you in social ties, who are also clumped together with one another in space, indicating that they're represented similarly to one another in this brain region. We can also test if and where brain regions respond such that distinctions are based on how well connected people are when we're seeing them. And so if this were the case in any brain region, that brain region's information signature would look something like this, where the well-connected people are close together at the top here, indicating that they evoke similar neural patterns to one another, and they're far away from people who have few connections in the social network, who are down at the bottom here, indicating that they're encoded quite distinctly from those people. And what we found was that there were brain regions that seem to sort people out along each of these lines. And so here, the colors overlaid on the brain signify what seems to be reliably encoded in a given brain area. So areas colored in an orange encoded who's really well connected in the social network to well connected others. So eigenvector centrality, in other words. Purple signaled social distance, so degrees of separation away from oneself. And green areas signified the extent to which someone bridges between disconnected people or groups, also called constraint. And in this data set and in follow-up work, we've done analyses where we control for things like personality traits, appearance, demographics, and so on. And so it really seems to be this knowledge of where someone sits in your social network that's driving these results. And so these results tell us two key things. First, they tell us that when we encounter people who we know, our brains do seem to spontaneously encode where those people sit in our social networks. And second, based on our knowledge of what else these brain regions do, they can provide us with testable hypotheses about how this information about others' positions in your social networks, once it's encoded by the brain, might shape how you process further information and how you think, how you feel, and how you behave. So for example, information about who's really well connected is carried in brain regions that are associated with, among other things, thinking about other people's thoughts. So these are some of the brain regions that I told you were involved in social cognition at the very beginning of my talk. And this suggests that being really well connected to well-connected others might impact the extent to which we pay attention to what other people seem to be thinking. And in some follow-up work, we're finding support for exactly this idea. And so a lot of the work since we've obtained these results has focused on questions like, how do we acquire this knowledge? So how do we learn about the structure of our social worlds? And what do we use it for? So how does this knowledge shape how we think, how we feel, and how we behave? And so now we've seen at least one way that integrating the study of social networks into neuroscience can tell us about the brain. So it seems that part of what our brains are doing when we're making our way through the world and encountering other people is tracking and encoding information about where those people sit in our real world social networks. But what about that other side of the coin? So what can we learn about our social networks by studying the brain? So in the remaining time that I have with you today, I'm going to shift attention to what our brains can teach us about our social networks. And in the work that I'll focus on, we tested if how our brains respond to the world in general and broader characteristics of our brains are predictive of where we sit in our social networks. So many people have this intuition that we're friends with others who are like us, or that birds of a feather flock together, as the saying goes. And this is not a new intuition by any sense. It goes back at least to the 1500s, and sociologists have confirmed this. So across cultures and contexts, humans tend to be surrounded by others who are like them in terms of a whole range of factors like age and gender and so on. And this tendency is called homophily, which means love of the same. So it's been suggested that homophily might benefit us because it facilitates collective action and cohesion and empathy, since similar people are more likely to share assumptions, goals, and experiences in common. But this kind of explanation hasn't really been tested directly. And in the same vein, you might have the intuition that your friends aren't just people who fulfill certain demographic criteria, like same gender check, same race check, and so on. Within certain groups, we of course resonate with some people more than others, and you hopefully have friends who fall into different demographic categories than your own. But until recently, a lot of evidence for homophily has really been based on pretty coarse variables like demographics. And this is despite the perception that a lot of us have that our friends are people who we sort of click with, who respond to things like we do, and who see the world from a similar perspective. So here we want to test this notion. And one window into our thoughts, our emotions, how we allocate our attention, and how these processes unfold over time is neural response time series to rich dynamic stimuli. So some really elegant work by Yuri Hassan's group and others has shown that if you take a set of people and show them an engaging dynamic stimulus like a movie, and then extract neural response time series from corresponding brain regions as they watch that movie, that stimulus, they become sort of locked together in time. So it's just sort of a cartoonish depiction. It's not real data, just to show you conceptually what I mean. And the similarity of responses, depending on where you're looking in the brain, of course, reflects similarity in things like their interpretations, what they focus on, their emotional reactions, and so on. And so it follows that if we are exceptionally similar to our friends in how we attend to, interpret, and respond to the world, we'd expect neural responses to be exceptionally similar among friends. And so to get at this, we again characterize the full friendship network of another graduate program We had a subset of students come in for an fMRI study where they just watched a set of videos. So again, those people are colored in peach here. And these videos were selected so that people would have been unlikely to have seen them before, and so that they were engaging and would recruit many of the same cognitive and emotional processes that characterize our everyday mental lives, and also to minimize mind wandering, which would, of course, be a source of noise for us, stimulus unrelated thought. But we also want to choose things that would foster some meaningful variability between people, because different people might interpret them differently or pay attention to different parts of them, or might emotionally respond to them differently. So we chose some things that some people might find really sweet, touching, or adorable, but that others might find kind of sappy and cloying and overly sentimental. We had other things that might appeal to people with a certain sense of humor, but not others, who might fail to appreciate or even detect the humor in some cases. And we also use some things that might be understood or parsed differently depending on one's background knowledge and interests, and also arguments that some people might really resonate with, but that might evoke skepticism or even contempt in others. And just to give you a sense of our participants' experience, I'm going to show you about 45 seconds of some brief excerpts of some of the things that participants saw on the scanner. And so in the interest of time, each of these video excerpts has been trimmed to just be like a few seconds long, but in the actual study, each video clip was at least a minute or two long.
When I was outside on my first spacewalk, I was on the dark side of the world, over the Indian Ocean.
And by having workers who only had to do one thing, they could pay them a low wage, and it was very easy to find someone to replace them.
The water squeezes out of the cloth, and then because of the surface tension of the water, it
is insulting when a coach is making 5 to 10 to 15 times more than a college president.
These babies are not strong enough to cling, and they fall to the ground repeatedly.
It's at times like that you don't often think of funny things to say, but I did. And I turned to the rescue workers and I said, "Talk about a rough day at work." That was referring to spending the time up there being pretty rough. Talk about a rough day at work.
Okay, so again, in the study, each clip was much longer than that, usually a few minutes each, but that was just to kind of give you a taste of the kinds of things that our participants would have experienced while lying in the scanner. And while participants watched the videos, we measured how activity across the brain rose and fell over time. So on top here, you see data from 2 participants that's been sped up a bit to give you a sense of the data without having to linger too long on the slide. And on the bottom is activity summarized for the circled brain area for these 2 participants. So these people look pretty in sync with one another. They also happen to be friends. And we want to test if and where, in general, people who are friends and who are closer to one another in the social network would have more similar neural responses. And I'm just going to show you some descriptive statistics to give you a sense of the overall pattern of results. And I should note that this is controlling for similarities in all the demographic variables that we could get our hands on. So here, warmer colors will mean more similar than average for a given brain area, and cooler colors will mean less similar than average for a given brain area. And when you look at people who are 3 degrees of separation from each other, so friends of friends of friends, people are responding relatively dissimilarly to one another as reflected in these cooler colors. But as you get kind of closer together in the network, you see warmer colors with friends responding to what they're seeing exceptionally similarly. So this is particularly true in regions that determine things like how we allocate our attention, like the superior parietal lobule, which is circled here, and also areas involved in high-level construals of events, so kind of our interpretations of the world, like the angular gyrus, which is just below it. So this is consistent with the idea that we do tend to be surrounded by people who process the world similarly to how we ourselves do. And so to return to that earlier question, do we respond to the world like our friends do? It seems that we do tend to be surrounded by people whose brains process the world similarly to our own. And so now in ongoing work, we're doing additional research looking at longitudinal studies and using new paradigms that allow us to ask questions like, to what extent do we associate with people who are already like us in how they respond to the world? And to what extent do we grow similar to and sort of align ourselves with our friends and the people around us over time, and in what ways? And while this work is still in progress, we are seeing that we can predict who will become friends with one another and who will end up close to one another in social ties from brain data that we collect around the time that people first meet each other. And on top of the work that I've just told you about, that's focused on how your brain processes information about the world around you, in other work, we're finding that we even see similarities among friends and people close to one another in their social networks when they're not seeing really anything at all. And so we each have our own distinctive functional connectome that just characterizes how different parts of our brain are coupled at rest. So when we're not doing much of anything. And these can be thought of as sort of connectivity fingerprints, so to speak. And they're unique to individuals, and they're also related to individual differences or trait variables in various ways, including things like how people tend to think and to behave. And we've recently found that our functional connectomes, so the patterns of connectivity in each of our brains at rest, are also particularly similar among people who are friends and who are closer to one another in social ties, especially among people who live close to one another and can interact with one another readily, as you can see here by this red line that's steeper than the rest. So this data was collected on residents of a small village on an island in South Korea. So my graduate student Ryan Hyun and I collaborated with our colleagues there, including Yusik Yoon and Jin-Young Chae on this study. And so we have participants' brain data. We also have their social network data. And we also know where they live in the village. We can map out how close or far their houses are from one another. And we see that even after accounting for things like demographic and personality similarities and all the things that we typically measure pretty readily with self-report surveys, people with more similar functional connectomes tend to be closer together in the village's social network. And this relationship between social network proximity and neural similarity is particularly pronounced among people who live closer together, which makes sense if you consider the fact that, for one, people who are closer to one another in both space and social ties, they might have more chances to influence one another. And also, they might be more likely to end up in common situations and common environments that might also influence them both. And you could also consider the fact that people who live far from you might be a mix of people who you aren't compatible with and people who you might be better friends with if you live closer to them and got a chance to know them better and could sort of, befriend people who you're compatible with. So all these factors could contribute to this relationship between social network proximity and neural similarity that we see being more pronounced among people who live close to one another. And so hopefully what I've shown you today is that we can fruitfully combine these levels of analysis. So looking at information processing within individual brains and the social networks that people inhabit to learn things both about our brains and about our social networks. So for example, we see that when we encounter other people, even when we're not trying to, and even when it might not feel like we're really doing much at all or processing any information at all, our brains automatically retrieve information about where those people sit in our social networks, and this likely helps us prepare to think and behave appropriately. And we also saw that people tend to be surrounded by others whose brains respond to the world similarly to how they themselves do and who have similar functional connectomes. And so I think that by combining these sorts of traditionally siloed approaches from psychology and neuroscience on the one hand, which tend to focus on the individual, with methods for characterizing our patterns of real-world social relationships on the other hand, we can open up a lot of what I think are really exciting questions because, if we really want to understand social thought and behavior and their neural basis, we can't just treat individuals as isolated units or even as dyads or pairs of people and expect to be able to tell the whole story. So even when we're just looking at two people having a conversation or interacting in some other way, what's really going on under the hood, both in terms of how they're perceiving and reacting to one another and how they're influencing and being influenced by one another, seems to take into account this rich fabric of social ties in which each one of us is embedded. And so with that, I just want to briefly thank the many people who've been involved in this research. So my lab members, Alyssa, Ruby, Mung, Brian, and Miriam, and our collaborators, especially Talia Wheatley and Adam Kleinbaum at Dartmouth, and Yusik Yoon and Jin-Young Chae at Yonsei University and Seoul National University, and our funding sources. And of course, thank you for your time and attention. And I'm looking forward to chatting with you during the discussion period.